Hi everyone, so in this video I'm going to cover fermentation and sort of the importance behind it and also how it's different from anaerobic respiration. So yeah, to start I'm just going to draw sort of like the overall idea behind fermentation. So this is likely something that you've seen in class, but we know that glucose can be converted to 2-pyruvate molecules through glycolysis. But then really the issue here is that we no longer have our terminal electron acceptor, right? So fermentation is occurring when we don't have O2. And oxygen was normally our terminal electron acceptor for the electron transport chain. So we can no longer do oxidative phosphorylation. And so instead, this is sort of what the body will do, basically. And so in this case, what we're going to do is pyruvate will undergo this process of fermentation to become lactic acid or ethanol, depending on what organism we're in. So humans will convert to lactic acid, and in yeast, like when we're making beer and stuff like that, that's conversion to ethanol. But the most important part of all of this is the fact that this allows us to regenerate our electron carriers. So I'm just going to draw this and then I'll break that down. But what I've drawn in red here is really the important thing, right? So glycolysis is not that bad, right? We're getting 2 ATP, net ATP for every run of glycolysis, but we also accumulate 2 NADH molecules, right? So eventually, if we didn't have fermentation and we just ran glycolysis forever and ever and ever, we would run out of NAD+. So the oxidized version of NADH that is available to accept electrons. So if we didn't have fermentation, glycolysis would only be able to run for a certain amount of time. Basically, like at some point you would have all of the NAD+ in the reduced state because glycolysis is reducing them. So they would all be in their NADH state, and you would have no more NAD+ left over to allow glycolysis to keep going, right? Glycolysis, one of the inputs is NAD+. So if we remove that input, the whole of glycolysis can't work. Like you can't get from glucose to pyruvate. So fermentation really allows for this alternate pathway to get rid of these electrons when we don't have oxygen. But obviously, all of the ATP that we made earlier in cellular respiration was mostly through oxidative phosphorylation. And so now we don't have that anymore. So we will lose a lot of ATP. And you can actually mainly see that in the fact that our end products here are still in a partially reduced form, which means that they still have electrons in them that could have been harnessed for energy, but they're wasted in this case because the body just hasn't evolved in a way that it can do this in the most efficient way, right? This is really just more of like a backup plan sort of survival thing. So really the main idea here is, well, we'll make as much ATP as we can, but we have to make sure that we can regenerate NAD+ so that we can keep doing glycolysis to give us that ATP. So we don't really get a lot of ATP, and we also don't get all of the energy that we can out of these molecules because we're still leaving them in their partially reduced forms. So that's really the main idea behind this. And the other thing I want to mention is in this case, pyruvate is really what is becoming our terminal electron acceptor, because the NADH is dumping its electrons onto pyruvate to then oxidize NADH to NAD+, and pyruvate is becoming reduced, right? We have to remember when redox reaction happens, something is being oxidized and something else is being reduced. So pyruvate is really acting like our terminal electron here, or a terminal electron acceptor in this case. That's for humans in ethanol, in like fungi and plants. It's actually a modified version of pyruvate called acetaldehyde, but it's effectively the same idea that a version of pyruvate is acting as the terminal electron acceptor. So hopefully that gave a good like outline of what's sort of happening here, but really like the main idea is that we're unfortunately not making as much ATP as we were before, and you can see that in the fact that our products are still partially reduced in that we haven't extracted all of the electron energy from them as we could have. And so we sort of see that in the fact that we're not making as much ATP as we were when we carried out all of cellular respiration. And one last thing I'll mention is that this is very different from anaerobic respiration. Anaerobic respiration occurs in organisms that are otherwise like used to not not having um, oxygen an oxygen supply. So they've sort of evolved a different cellular respiration. It's similar to cellular respiration, but their terminal electron acceptor is some inorganic molecule. So they're still going through a decent like the full like cellular respiration pathway. They're making a little bit less of ATP than normal like aerobic cellular respiration, but they're making a lot more than fermentation. So they are very different. And fermentation is, in a sense, it really cuts off how many of those steps of cellular respiration we're doing. We're really only doing glycolysis, and then fermentation is allowing us to regenerate NAD plus to continue doing glycolysis. But anaerobic respiration is really not that. It's going through a full like cellular respiration pathway, but at the end of the electron transport chain, we're just using a different terminal electron acceptor. So hopefully that also cleared up the difference and just gave an idea of what's going on here.